The first one is Bacillus anthracis, and as the name suggests, it is a cause of anthrax. So the anthrax causing bacteria. So we had this as a part of a lecture on bioterrorism. I have to update that lecture because there's a list of categories by CDC and the government. Where do they put a particular microbe in, like category A, B, C, D? So A is like more serious. So that needs a little bit of update. So we'll see how do we do on that. It should be seven, whatever you see on the top. Lecture number 7, PDF, Bacillus anthracis and Corynebacterium diphtheria. It's not a PowerPoint, it's a PDF. No. It's not. That's what I'm saying. It's not. So you have to look for, I think it should be there in the first, where you have the recent lectures as, where you have immunology lectures. In the end of immunology lectures. You find it there.
It's under where it says slow micro.
Oh, it did go over there? Okay, there you go. All right, let's get started. And so you can see from here, it is categorized as Group A. And Group A category in terms of agents used for bioterrorism. And as far as the cause for anthrax, again, is a gram-positive. So we are talking about gram-positive so far. It has a capsule. It forms spore. It has rod-shaped. So bacillus. And then again, as you can see that this was a big scare a couple of years ago. And anything that forms spore is dangerous because you can put it in a powder form and post it, as you saw in those instances. It does have at least, in theory, at least 4 different forms of disease. And you can see the most common is skin or cutaneous, as has been for staph, strep, and Clostridia, which also are skin and tissues. Very rarely, it can have respiratory tract where you can inhale. You can inhale the powder or inhale the spores. It also has some documentation for gastrointestinal disease, and oropharyngeal is the least common. The things for many of these infectious diseases, so to speak, that you need to be familiar with, that how is it transmitted and what are the chances of us getting it and how can we protect and prevent ourselves as well as patients from there. So you can see over here, you have to have a direct skin contact with the spores. And the other way it can spread is that we can be a part of a process where we can get it from either infected animals, if you're working in the farm or tend to visit the farm, or you come across any animal product. Because it's a very common immigration question as well when you come at land at O'Hare Airport, have you visited a farm? So that's where it is coming from. There are some diseases that we pick up because zoonotic diseases. These are diseases from the zoo, zoonotic diseases, especially in European country because there was a big scare for the mad cow disease, especially for the beef. So you can see from here, respiratory again, and very common practice for some of the cultures, people consume undercooked or raw meat, and that also is known if there are spores in those meat processes that can give you anthrax. As far as report is concerned, many diseases are, and again, I would want you to make a list as we speak, notifiable diseases. Some of the diseases are notifiable diseases. You have to notify the government. So that's important. You have to notify the government, and then they have to make sure, because of their nature of being infectious, and some of the diseases will want you to notify to local, state, or government agencies. And for the physio part, or the structural part that we've been talking so far, is that they are aerobes as well. So they would want to survive where there's rich oxygen. They are also found in soil. As I said earlier, cattle are routinely vaccinated. So we vaccinate the cattle so they don't get anthrax. And these are the drug of choices that we have. And as I said earlier, you can get it from a minor cut. So you can get it from a minor cut in terms of their transmission into the system. They do carry mortality, and again, that figure may not truly reflect the right incidence, because it doesn't tell you mortality in which age group, for example, mortality in which state, which country. So there are a lot of variables there when we say of mortality. So that term used over here will give you an idea as to everybody who gets anthrax may not die from that. So it's 20% is like 1 in 5 in terms of person who get it. Okay. Now, the other important thing that we've been talking about earlier, and those of you who came for gram staining, we were talking about minimum inhibitory concentration, MIC. Or are these both doors locked? Yeah. Why don't you just open? I don't know how to. Because I saw when this door was open, whole floor was resonant with Kumar's voice. It's like strong. So we couldn't even study in our offices. So I just wanted to make sure that we don't cause botheration. So you can see from here, portal of entry in terms of infectious dose. So infectious dose, that if you are given a infectious dose, how much is it that you require to cause a disease? So you can see from here, the portal of entry has to play a major role. So you have to have 10,000 to 20,000 endospores to cause respiratory anthrax. As compared to ingestion, you have to have millions or 250,000 of endospores. But at the same time, you just need 10 to 50 endospore to cause skin. So that was a scary part, especially for that letter-related threat of anthrax. So you can see that is a real information for us to be particular about. Now, Department of Defense, that's what we say bioterrorism, not only CDC alone, but Department of Defense and WHO and UNO, all bigger organizations also keep a record of what is happening and so on and so forth, and a lot of geopolitical things as well. For example, you can see here, say believed to, Department of Defense statistics, they say that believed to possess anthrax weapons. So you can see Russia, China, Iran, Iraq, Libya, all these countries, according to Department of Defense, have stockpiles of anthrax because it was considered, or probably still considered, as a weapon of mass destruction to eliminate your enemies. So you can see, also keep in mind that if they are not particular about those stockpiles, you can get infected. and there can be a breakage of that. There are many things that can happen, but you can see from here, even the story that we were talking about earlier, they wanted to make sure that they can type that particular microorganism. For example, the microorganisms that we give you for gram staining, they are coming from American Tissue Culture Center, so they have a type strain for that. So we know for sure if we give you mycobacterium, it's not going to cause you tuberculosis. It is benign or healthy. So they have a tissue typing of each and everything. So our lab over here would not be permitted to use the real anthrax because we don't have the facility to do that. But probably University of Chicago, USC, or any other place may have that. So if you're working over here, if you come across that, you have to be more kind of vigilant of that. Okay. So you can see that was something which is there in terms of talk about the culture collections. And then again, some of things may be speculation, nobody knows, but this is for sure that those of you who are interested, the one used over here in this part of the world was coming from a very specific lab by a very specific person. And those of you who followed it up, I think he committed suicide, so we don't even know what happened afterwards. Anyway, what we are interested in this model is that if you are in a system, in an ecosystem, where you share the airspace, for example, this airspace is shared by people, this 90 people, couple of hours every day, right, for X amount of time, or Chicago. So all these things that we come across and they get infected or they carry infection can be a source of us acquiring that. So that's what, there is an interplay of transmission process, especially for the parasitic diseases. They're very complicated. When we'll teach you parasitology, you will see that we'll show you very complicated cycles of eggs and larvae, and then mature, and so on and so forth. But you can see that this is a cycle of anthrax, that vegetative forms can come from animal decomposition if an animal has died, and then again you can consume meat from the animal. If you come near them, you can also leave it on the spores in the farms. So there is a typical ecosystem, and also theoretically, a biting fly can carry particular endospores from animals to human beings, especially those who are working. That was the story of the swine flu that, if you remember, a couple of years ago, those people who were working in the pig farms, they acquired it and then passed it on to the people, and so on and so forth. But the point over here is that you have to understand that even with the best of all the healthcare that you may have, we still are living in an ecosystem. So we are vulnerable, so to speak, in terms of whatever is going around, and whatever meat that you're consuming, so on and so forth, in terms of some of the diseases that are there. Now, when we talk of this type of cycle, we also talk of a term, we call it a primary host or secondary host or accidental host. Now, some of the diseases are purely zoonotic diseases or diseases of the veterinary diseases. They are purely for that, for like bird flu. But again, since we consume, we come across, we use animal product, there are always chances of us acquiring it. So you can see from here, for example, these are the spores that can contaminate even the hide of those animals like cattle, sheep, and goats, and they can also ingest, right? And they can leave it in the atmosphere, in the, what you call, soil. And then again, those people who come in contact, for example, you come in contact with the animal product, that could be a hide of that particular animal, wool or hair. Again, is associated with mortality. Most of the time, is ingestion of contaminated food product, as I said, for any cattle. Again, is very high, 95%. And then again, some of the diseases are there in the dust. We call wool sorter's disease. Again, is a very high incidence. So you can see there are a lot of government agencies other than health agency or Department of Defense, even food, FDA, they also have to regulate quite a few things in terms of us not getting our, skin being exposed to all these things, and then some of the places we share, like, airports, train stations, and many things are common where people will come, and you can always have that threat in terms of acquiring those infections. So Bacillus anthracis is considered as a very high a suspect in terms of causing that kind of a panic at global level. Now, having said that, I'll just give you a brief idea of the ecosystem that we talk about, and then again, we'll discuss that again in detail as we move along for those respective bacteria. But the whole point over here is that, so if this is you, so you come across all these ecosystems, there are worlds around us. So you cannot sit idle. I want to worry about, for example, anthrax there in animals, but you consume milk, right? So you consume beef, meat, whatever. So you always have a contact with that, okay? Then again, you come across these insects or pets or some other small rodents or some pets as well that are there in the system, and you would never know they share the same soil, same field, same garden, or that habitation, and then they can leave all these things in terms of causing direct contact on the skin wise. Then again, in this case, I mean, the examples, if I was to give you like 4 different examples of 4 different infectious diseases, this is a typical for the anthrax, and this is a typical for plague, right? And this is a typical for tularemia, you can see from here, and this is again type A and type B. So you can see that all those animals, and you can argue there's no way any civil society or city or anything of that nature can particularly eliminate all these kind of factors. There's no way. Okay? So you can understand from this that we always are exposed to different factors that are there to cause disease. Okay? And then again, many of these basically are dealt by specialists. They're not even dealt by us, because I know it's very confusing. It's not easy. So there are specialists who will deal infectious disease specialists. They deal with all these things. So if you do plan a visit or a trip or whatever, hiking trip, so these are some recommendations that they want to make sure there may be an incidence of that particular disease in that particular area. So you always be vigilant for that. Okay, now I'll just concentrate on bacillus from this side. Don't worry about plague. But you can see from here spores being inhaled, right? And then we get exposed to the product. So you can see from here that a rabbit or rodents or rats or even pets, they basically can inhale those spores. They can go around and bring it to your home or bring it to you if you ever have those kind of pets, or if you plan to visit a zoo or a plant. So this is a factor that's always there. Other than simple factor that you are either consuming animal meat or animal product, may it be a hide of an animal, may it be, because they're spores. And I can assure you that no hide would be autoclaved. There's no way they're going to autoclave it and give it to you, hey, this doesn't have any spore. So you always have that risk, though relatively low, but academically you need to know that, okay? Now, how do you diagnose anthrax? Again, as I said, it's a notifiable disease, so it's a big scare, and what they normally do is that most of them present as skin lesion, so they want to aspirate or swab, and then do again the gram staining and culture, and you can do some of the, as I said, identification of that particular strain and isolate, because you want to trace it back to the origin. Okay, so that's important because, for example, none of us have ability, and I'll talk about that when I talk for bioterrorism, that how would people have to have that minimum knowledge how to convert a particular bacterial culture into spore. If you don't have that, you cannot be putting it in the mail and sending it to people. Right? So there has to be a person, a specialist person. Things are not that simple as people think they are. So otherwise, it's going to be very crude. So we want to isolate and identify that. Now, there are a lot of lesions that you normally see. I'm going to show you some of the pictures. You can Google it. You'll find it over there. If there is a inhalation of endospore, very high mortality. If there is ingestion for that in the food, is a 50% mortality. By the end of the day, you basically see a skin lesion. So you see a skin lesion. That's what pretty much very common is. You call cutaneous anthrax. That's what you normally see because you would not be able to see inhalation because it's in the lungs. You can't possibly look at the gastrointestinal anthrax because you can only look it up postmortem. But if you look at the virulence factor test, it's a capsule. Again, it is antiphagocytic, and most of the time it's the immune response that sets in and causes problems. And there are a lot of different versions of cutaneous anthrax available. It can affect any part of your body, but you can see the nasty looking lesion. And then again, the problem with these lesions is that they would not heal. Unhealed lesion is a major scare in terms of that. The other thing is when the physicians looked at it, they basically see that a progression over time. So you can see things don't come to that eschar, black colored eschar right away. So it has to start up with some kind of very small injury, vesicle formation, and then over time, you can see it really goes into real eschar as we see as a part of cutaneous. It can also be very ulcerative in terms of the pathology that we have. Some of the lesions, like if you've seen bed sores, but bed sores again are not that deep, but these lesions can be very penetrative, and they can see from here they like cut like a sever by a sharp knife. So you can see it actually goes very deep in terms of anthrax. Okay. And again, as I said earlier, most of the time is skin. There's no one format. It depends upon your immune response. It depends on many different things. So people have seen different aspects for that. And some of the New England Journal of Medicine, you can see some of places like in Africa where they are continuously living with those kind of endospores, and even the kids, children may be exposed, and they show all these type of different cutaneous lesions. It's treatable, we treat it, and then the healing process, pretty much, as we talked about, the repair process in terms of a chronic wound being repaired. So that's pretty much the case that you would see. This is again an early lesion. You can see a lesion with edema of the arm. And then again, I think I must have told you many times that all the skin lesions look alike. They all look alike. It's very difficult to figure out whether it is difficult to diagnose. So most of even physicians will refer it to skin specialists. They all look alike, but the treatment may be difficult or different. Okay, so this is like a post-mortem thing that you look at the necrosis of intestine after gastrointestinal anthrax. Treatment, as I said earlier, we can prevent it if that's possible. Otherwise, penicillin is there, as I said, for a preventative measure. We want to vaccinate the animal herd. And if the animals die, we want to take care of their carcasses. And then again, active immunization is available for some of those high-risk people. Okay, so that was pretty much a kind of a general information that would come into those 5-6 categories that you need to know in terms of bacillus, anthrax, And then again, I'll talk a little bit in detail when I talk about the response in terms of it being a bioterrorist agent. The other gram-positive bacteria, again, is Corynebacterium diphtheriae. Again, is a causative agent of diphtheria, because I just put up one of the common diseases that you normally see. They are gram-positive. Pleomorphic means that sometimes the rods are rods, like stakes. Sometimes they are club-shaped. Sometimes they are swollen on both sides. So they kind of change the form. So we call those kind of rods pleomorphic. The most important thing for this particular bacteria is that, remember I told you that some of the bacteria per se may not have this ability to cause damage on their own, but most of the damage that you see is by the toxins that they release, especially diphtheria. It's called exotoxin. We call AB exotoxin. And this is the one that inhibits protein synthesis or causes tissue damage, right? And again, like Bacillus anthracis, it also has respiratory and cutaneous forms. We talked about this figure over and over again because the protection that we have for all environmental sources of bacteria again is skin, right? And then mucous membrane. And this actually is a typical slide I picked up for one of the textbooks, Sherry's Medical Microbiology. And you can see from here, again, lungs, gastrointestinal tract, urogenital tract, they're all being lined by epithelium, or mucous membrane. So mucous membrane and the skin. In this case, you will see that bacteria actually goes into the lungs, and then toxin is absorbed. So when you have the Corynebacterium, and then it releases that green color, this toxin, that's diphtheria toxin, and it can spread. Even if it spreads through the blood, it can go where the blood takes it to, especially in the heart, causing myocarditis. So that's also one of the things that you have to keep in mind. A sore throat. Remember when I said sore throat, if I ask you, name some bacteria which cause sore throat, but they lead to diseases of the heart. We talked about Staph pyogenes, right? That led to the rheumatic heart disease. The other one is Corynediphtheria, because of the toxin, it goes and causes myocarditis. All right, now how would you acquire that? So in the previous picture, you said what was the name I told you, type of infection, that when people breathe out or cough out or sneeze, that kind of transmission, what is it called? What kind of infection is this when you acquire infection through other people, coughing, sneezing, talking? There was a term that I used, yeah. Droplet infection, droplet. Because the good from the droplet is that when people sneeze, is a little bit of water vapor that kind of attaches and covers it up. So it's very protective. So this is called a typical water droplet that actually goes and causes infections. Now, as I said earlier, even if the bacteria which goes into the respiratory system, and it's not invasive, bacteria is not invasive, it doesn't have ability to adhere and penetrate the mucous membrane, but it can release a toxin that's going to play important part in this case. So that toxin is absorbed. So bacteria will attach to your throat, release the toxin will be absorbed in the blood, and then bloodstream will take it to different organs, particularly muscle of the heart, causing acute myocarditis. So that's the disease that this particular bacteria produce. Again, you can see from here, this is C. diphtheria. It basically attaches to the receptor on the skin, on the mucous membrane, especially of the throat. And then again, it's going to be phagocytosed. And then you have a toxin, which is A and B toxin. So you can see these toxins will go into the system, destroy the tissue, and then again will be absorbed into the blood, and then will be taken up by blood to different parts of the body. Now, remember, I talked about pseudomembrane. Which was the bacteria I talked about causes pseudomembrane? It was probably yesterday, correct? Pseudomembrane, we talked about C. difficile, C. difficile, antibiotic related diarrhea, right? So the other pseudomembrane that is very common that we normally see in this patient is pseudomembrane caused by diphtheria. So if you look at the throat, you'll see the pseudomembrane. But that pseudomembrane we talked about, the Clostridia, was in antibiotic-related pseudomembrane that was in colon. Now, if you remember when we talked about these toxins, and especially for their ability to inhibit protein synthesis. So the whole point for this toxin is it gets absorbed and enter the cell, and then again, one of them basically catalyzes the reaction that we have ADP ribosylation to EF2. This process over here, where ADP ribosylation to elongation factor 2, that's what we say, elongation factor 2, will inhibit the protein synthesis. So you can see that happens at the level of ribosome. So again, keep that in mind when we were talking about the antibiotics acting at the ribosomal level. We want to stop protein synthesis. So you can also see that we can learn from a bacteria how a bacteria blocks protein synthesis. So we can also use that technology in real time and make a drug or antibiotic that can help us to achieve the same. So the point I'm trying to make for this particular bacteria is that bacteria does not need to invade. So the pathogenic or virulence factor for bacteria may not be adhesions, invasions, and so forth, but it is the diphtheria toxin that is released and absorbed into the bloodstream. This is a typical gram staining over here. You can see seed Corynebacterium diphtheriae. And the other thing added to that particular exotoxin, remember I said that there are two important things in terms of toxin. One is an endotoxin and one in an exotoxin, right? And exo, I gave you a mnemonic. Exo is something that's secreted, exits out, as compared to endotoxin that's going to be a part of that particular bacteria. So this basically is an exotoxin. And this exotoxin has a gene that is called tox gene, right? Now, if you remember the sentence over here, when we talked about how do bacteria get antibiotic resistance, right? So in this case, if a Corynebacterium diphtheriae is infected by a virus, which is a bacteriophage, you can see bacteriophage, and this bacteriophage will pick up that particular tox gene from there and insert it into another bacteria to cause the damage that this bacteria causes. So that's where it's like transfer of a weapon from one bacteria to another set of bacteria by a process. What was the name of this process in terms of genetic transfer? What are the different types of methods that the resistance or the DNA gets transferred from one bacteria to another? Conjugation. Okay, very good. Transduction and transformation. Is that correct? So which one is this one? Okay, I'm going to leave it on you and ask you some other day. Keep in mind, I'm going to ask you this question because this is an important aspect where we find out that bacteria is able to pass that information to another bacteria to cause damage. Okay, now, if you look at the whole process, of that particular gene that we talked about in terms of causing damage. So what you will see is that that particular gene has a capability to cause protein lysis. That's what it is. We call it proteolytic one. So if you look at the sequence over here, you will see that it has two molecules, A and B, and they attach by a disulfide bond. And then again, one of them is, as I said earlier, is for binding, and the other one is the one that is going to cause damage. So that kind of in a process where a toxin has two components, we call A and B. One of them is for binding, and the other one will cause the real damage. We call that process a classical AB toxin that is very kind of particular with Corynebacterium diphtheria because of it causing diphtheria. So we want to come up with the drugs that will block that capability of attachment of that toxin to epithelium so we can prevent the damage. If you look at this epidemiology, people can carry. So you can carry a bacteria as a normal flora, but you yourself may be asymptomatic, you don't get disease, or you can pass it on to the other people. We call those people carriers, and they can give it to the others. Humans are the only known reservoirs, so this means we cannot acquire it from animals because they don't keep it. And we keep it in our oropharynx or skin. Spread from person to person is respiratory droplets or skin contact. And then again, we all get vaccinated, you know. So we all are DPT, vaccine diphtheria. So that's the common vaccination that everybody's protected from. And then that's why we see very uncommon in United States. But having said that, what they found out recently that immunity was lagging in terms of long-term protective immunity with diphtheria, and even we see this coming back. So that's going to be a big problem again, even after the success story of the childhood vaccinations, things are coming back. So they basically are arguing whether it is a failure of that particular vaccine or there is a genetic transfer or a pathogenic strain in terms of the newer versions of Corynebacterium diphtheria. So it could be both, right? So they over time have come up with a plan that we do see that, but the incidence of, we should have no incidence, that's why it's uncommon, but again, it has come back. So most of the time we see in terms of sore throat, and people have low-grade fever. And what is malaise? Malaise is like a feeling that you get that something is wrong with me. It's unwell feeling. You kind of feel, before a disease. We call it malaise. So you know that you're not normal. Something's wrong with me. I'm feeling sick. So that kind of a feeling is considered as a malaise. They usually get it before viral infection. So you don't feel like doing things. So that's kind of a prodrome that you normally see. Then you look at a pseudomembrane in the pharynx. If people become critically ill, they may have cardiac and neurological complications, especially in the kids. And that's for respiratory diphtheria. But again, you would not see it here. But if you were to go in some countries where there is no childhood vaccination, you definitely will see, especially in the younger children. Cutaneous diphtheria, again, is something like a papule. The point is non-healing ulcer. Non-healing ulcer can be infectious, can be cancerous. As I said earlier, it is something that you need to ask the dermatologist and get an expert opinion about. It's not that simple. So this is the pseudomembrane. You can see a pseudomembrane at the back of the throat of the child. Very common if you look at, just like we talked about the other day, strep throat. So the commonest cause of the sore throat is viral. But viruses, again, you don't need to do anything. But if it happens to be strep throat, especially for the strep pyogenes, which is group A streptococci, it is dangerous because it can lead to heart disease because of the cross-reactivity of the proteins that we talked about, okay, M proteins and F proteins. So in this case, what you see is that there is a pseudomembrane over here. So bacteria is attached, and then toxin will be absorbed and go into the system. How do you diagnose it? Again, look under the microscope. You have to perform a culture. And sometimes we have a very selective culture. What it means is that if you were to make them grow on a plate, they would not grow on agar, simple agar. They would not grow. So you have to add all these things as a growth factor, cysteine, tensile medium, colistin. So you have to add some things over there. Sometimes we even add antibiotics over there. So we want to make sure that it grows whatever we want it to grow. Not everything grows there. You can also do a biochemical test, especially if you want to do a gene sequencing. As I said like a minute ago, what was the name of that gene that causes the exotoxin? Huh? Tox gene. I just said tox gene. So that's a tox gene which will be taken up by the bacteriophage and be inserted into the other thing, tox gene. You can also do a PCR, polymerase chain reaction, or ELEC test to demonstrate exotoxin. So that's a typical example. If I was to ask you which bacteria causes exotoxin, of course, this is going to be number one on the list. And then, well, we will talk about endotoxin later. Okay, now finally, we need to do something. Again, the problem is that we have to, if a person gets infected, so we're not worried about giving antibiotics because we know the antibiotics are not that important. We want to first neutralize the exotoxin, right? So that's the first line of defense we have. So you want to give diphtheria antitoxin, right? And then, of course, we would also want to give an antibiotic so that they don't divide and make more exotoxin. And as I said earlier, diphtheria vaccine is administered as a part of a routine diphtheria, pertussis, and tetanus, DPT. So do you have a DPT vaccine or as a normal childhood vaccination? And why do you need to give a booster shot? Why do you need to give a booster shot? Well, this is a basic concept of primary immune response and secondary immune response, because if you get primary response, you just get IgM production, and the levels of IgM will go down. You don't want IgM, you need IgG. For IgG, you have to give secondary immune response so that IgG stays in the system forever. So this is the basis of vaccination. If you understand that you'll understand. Otherwise, you're going to just show a blank face to the patient. He's going to ask you, why do I need to have a booster vaccination? Well, you can say, wait a minute, let me Google and find out. But most of the time, the patient would have already Googled ahead of you. So they're just testing you. So things are tough these days because many times, even the patients, they ask you the question, they know the answer, they just want to test you and verify whether you know it or not. So it's a difficult time, but I wasn't expecting you not to know why do we give booster shots. But of course, because we want to have a second immune response, which is going to stay for a good amount of time and also produce IgG. Okay, so let me finish that and